I'm doing the intro.
All right. Good morning, everyone. Thank you for joining us on live stream. My name is MK, and I'm the program coordinator for the Headwaters Science Institute. We have had a wonderful time with all of these students the past 2 weeks doing research projects where they collected data at Rodeo Lagoon, and they're very excited to share their findings with you today. If you have any questions, please feel free to drop them in the chat, and I will show them after each presentation, and they can answer your questions. Thanks for joining.
Okay, hi everyone. Our project is called Dissolved Oxygen and pH Effect on the Biodiversity of Macroinvertebrates. Our goal was basically to explore how important water quality factors like dissolved oxygen and pH affect the variety of macroinvertebrates living in aquatic ecosystems, and we did this specifically in the lagoon in Rodeo Beach. Okay. So before we started collecting our data, we wanted to understand what past research said about this topic. So in past research, studies have shown that factors like pH, nutrient levels, and turbidity all play a role in shaping macroinvertebrates' diversity and abundance. And basically, when the water chemistry shifts, even slightly, it can potentially change which species can survive there. Another thing we found is that higher salinity levels usually reduce biodiversity because many macroinvertebrates can't tolerate super salty conditions. And we were really interested in seeing how this plays a role in our own testing sites. We wanted to know, does poor water quality actually make a noticeable difference in the number or types of organisms present? We also thought about human impact, how runoff pollution can change the pH or oxygen levels in the water and in turn affect these small but important species. And this research matters because macroinvertebrates are a great way to measure water health, and essentially they're used as bioindicators, meaning their presence or absence tells us how clean or polluted an ecosystem might be. So our main research question was, how does aquatic pH and dissolved oxygen affect the presence and biodiversity of macroinvertebrates? Our independent variables were pH, dissolved oxygen, since those were the factors we were testing and comparing. And our dependent variables were measuring biodiversity, including the Shannon Diversity Index, species evenness, and population size of macroinvertebrates, because those are the outcomes that change based on the water quality. And for our hypothesis throughout this, we predicted that water with a neutral pH and high levels of dissolved oxygen would have a greater biodiversity and larger populations. And this is because we predicted that extreme pH levels can dissolve macroinvertebrate exoskeletons, and very low pH can also stress organisms. And on the other hand, higher dissolved oxygen means there's more oxygen available to support life. And we also noted that high salinity can make survival more difficult since organisms need more energy adapting to those conditions. But overall, we expect to see the most diverse and stable macroinvertebrate populations in sites with balanced pH and plenty of oxygen.
All right, our testing sites. We collected data from 12 different sites around the lagoon, as you can see on the map here. We sampled 3 locations on each side so we could compare patterns in biodiversity and water quality under different conditions. For example, sites closer to the ocean with higher salinity had fewer species, whereas densely vegetated areas had more biodiversity. This one.
All right, so as Jonah mentioned, we went site to site. With this set of materials, I'll give you guys a second to just read them quickly. Some of the most important ones, I think, were the DNet, the pH and alkalinity tests, as well as the dissolved oxygen meter. And so we used those tools to go counterclockwise around the lagoon from each site, as Jonah mentioned. And we had one member of the group disturb the sediment with the bucket and then scoop the cloudy water filled with sediment, macroinvertebrates, and other things, pick it up, let the water drain out, and then he dumped it into a bucket. From there, at the same time, we had other members of the group sampling the water. So depending on the convenience, we either sample the lagoon or from the bucket with the pH and alkalinity tests, as well as a dissolved oxygen meter. With those, we did 2 samples per site just to avoid any errors that may be attributed to our measuring technique. From the bucket, I know that at the same time, we had other members of the group taking out the macroinvertebrates. So that's sifting through with pipettes along with spoons, trying to pick them out and sort them into the ice cube trays sorted by type of macroinvertebrates. And we used field guides to essentially determine which one was which so that we can make clear decisions. After that, we put all of our data into a sheet and moved on to the next site. And we repeated this 12 times. And then after our field day, we came back to the classroom and we used this. We made our data online, basically, converting it from paper data to online data. And then we made a series of graphs to basically understand what our data meant and how we can use it to further our hypothesis.
Yeah, so I'm going to quickly go over the results of our quantitative data between our independent and dependent variables. Most of our conclusions were not statistically significant, which requires a p-value of under 0.05. So this was our relationship between dissolved oxygen and macroinvertebrate biodiversity. Our p-value was 0.55, which is no evidence of a relationship. This is dissolved oxygen and macroinvertebrate species richness. There's also no evidence of a relationship with a p-value of 0.61, despite a very weak negative correlation with the linear regression line on the graph. This is dissolved oxygen and macroinvertebrate population size. And so there's an appearance of a small positive correlation on the graph, but a p-value of 0.51 means that there is no statistical evidence that there's a relationship. This is pH and macroinvertebrate biodiversity. Same thing, 0.6 p-value, so no significant evidence that there's a relationship, even though there's a weak positive correlation on the graph with the linear regression line. pH and species richness, same thing, 0.56, no evidence that there's a relationship. pH and macroinvertebrate population size, this was our least statistically significant graph with a p-value of 0.98. The linear regression line is also flat. But we actually, when looking at a quadratic regression line, there was a pattern. The statistical test gave the same result of no evidence, but this was something that we found to be interesting. So those are our quantitative relationships. And then actually we found one slightly statistically significant relationship between pH and dissolved oxygen. So the p-value was 0.07, so it's close to being statistically significant. And so as pH gets more basic, we found that the dissolved oxygen levels were higher.
Yeah. So although our group wasn't really able to observe like many quantitative trends in our data, we did observe some like qualitative trends throughout the day. We noticed sort of as we moved down the lagoon further away from the beach and towards the stream, the like types of species we were observing sort of shifted. Around the beach, we mostly saw only 2 species, which were aquatic isopods and scud. Those are shown like here. Yeah, and then, but closer towards the river inlet, we saw like a much greater kind of diversity of species, including this blood midge larva, these water boatmen, snails, and even like the coolest thing we found on the day, which was a dragonfly larva. Yeah. So in addition to this kind of trend on the species, we noticed a large visible difference between sort of turbidity in the water quality on both sides of the lagoon. So closer towards the beach, the water was a lot clearer, a lot less like stained, but more towards the river inlet, it was a lot dirtier, a lot more tainted. And this could possibly lead to interesting results if we tested like turbidity in the future. In addition, we probably would see a trend in like salinity and biodiversity, given like we saw a lot more diversity of species towards the river inlet, but again, we weren't able to test salinity because our meter broke. But that probably would also yield interesting results. So, yeah.
Yeah, for our discussion slide. So our results were mostly inconclusive. We couldn't find a clear or significant connection between dissolved oxygen, or pH, and macroinvertebrate biodiversity. Although biodiversity was slightly higher around neutral to basic pH levels, the relationship was still weak. The only somewhat significant pattern was between pH and dissolved oxygen, meaning they tend to rise together, but they still didn't translate to higher diversity. This means our hypothesis that higher dissolved oxygen and neutral pH would increase biodiversity was rejected because there wasn't enough evidence to support it. Instead, we noticed stronger patterns with habitat type and vegetation. Sites with more vegetation had a greater variety of species, whereas more exposed rocky sites near the ocean had fewer species. Overall, the results show that the habitat structure and complexity of ecosystems are likely more important for supporting biodiversity than pH or oxygen alone. Because of this, we had to accept our null hypothesis and reject our original idea that higher dissolved oxygen and neutral pH would lead to greater biodiversity. There just wasn't enough evidence to support that prediction. What stood out more was how much habitat type seemed to matter. Sites with vegetation supported a much greater variety of species, while the rocky, more exposed sites near the ocean had noticeably fewer. So even though pH and oxygen are important, our results suggest that habitat structure and ecosystem complexity might have a bigger impact on biodiversity overall.
Because of these results, we wanted to identify some possible sources of error. So we encountered a couple of systematic errors that would have affected our accuracy. So that would have came from like measurement or categorization. So one of our first ones was the dissolved oxygen meter. It took a consistently long time before settling on a number, which may have resulted in us consistently taking the data before it was even finished, which might have been a potential source of error. And then the salinity and pH meter stopped working at Site 5. And so we dropped the salinity data, but continued to take pH with testing strips and essentially change the medium. This may have caused an inconsistency in our data and basically affected our accuracy and precision. And another potential source of systematic error would be if we consistently recorded one species, adult versus adolescent, as two separate species.
Some sources of possible random error, which would have affected our precision, would have been usually in our picking out macroinvertebrates and categorizing them. So if when we dumped all of the sediment out, if we missed a couple, that would have affected our precision. Also, if we inconsistently scooped the level of water and sometimes got more sediment and sometimes got less, that would have affected our precision. And if we occasionally miscategorized one of the species, that would have been a random error that affected our precision.
Yeah, I guess kind of tying into that slide, some things, this slide has some things we can do in the future to improve our research. Firstly, we would probably take more samples. We only got 12, and I feel like with more samples, we could probably draw better conclusions. Yeah, another thing we could do is test our materials beforehand. As we mentioned, like our salinity meter broke. And if we knew that something like that might happen, we would know to kind of like pivot. And then also we should probably bring like a backup plan. So like an extra salinity meter, or like just know what we would do if it were to break. And then also scouting the research area beforehand would have been pretty helpful. Like it was, we found the terrain difficult to navigate at times. We probably lost a lot of time and data points we could have taken, like just traversing sort of the marshy area. And then some future questions we could consider that would be interesting. Like I kind of stated earlier, it would be interesting to focus on turbidity as a factor of like water quality, because we observed some like possible correlation there between that and biodiversity. And then another interesting topic to look at would be how the seasonality affects the biodiversity of macroinvertebrates. We know from like previous research that factors of water quality such as like pH and dissolved oxygen are highly variable based off seasonal algae blooms in the lagoon. So it would be interesting to view how those trends correlate to both size and biodiversity of macroinvertebrate populations throughout the year. Obviously, we couldn't do this in like one day, but that'd be an interesting thing to look at like over a long period of time.
So in terms of connections to the biodiversity EU, despite having inconclusive results, the framework and our hypothesis connected to the biodiversity EU quite a bit. The first connection was that we were measuring how abiotic factors such as pH, dissolved oxygen, and the proximity of the ocean affect macroinvertebrate biodiversity. And our hypothesis would have been true because the organisms need to solve the same problems of getting oxygen and water to survive. And so as conditions become more extreme, those that are tolerant to that change are the only ones who survive, which affect the biodiversity. The second point is that macroinvertebrate biodiversity is crucial to ecosystem health, which we learned through our literature review and preparing for the introduction. And so that connects to the part of the Biodiversity EU that states that biodiversity is key to maintaining adaptable Earth systems. And then the third connection is that because Rodeo Lagoon is close to a road, a parking lot, and a recreational area, it comes in contact with lots of human activities. And so human impacts such as runoff or pollution may affect the abiotic factors, which then in turn affect the biodiversity. And so that connects to the EU because it states that humans' actions alter natural systems and have an impact on the environment and the ecosystem. So.
All right. Finally, with environmental justice, our findings have broader implications beyond just Rodeo Lagoon. Protecting the environment and wetlands helps maintain clean water, nutrient cycling, and flood control, as well as runoff and pollution can disproportionately affect lower income neighborhoods near coastal areas and towns.
Yeah, I think the support of local ecosystems is really important as it can prevent erosion around the area, which in turn can support human life and ensure good quality of the life and access to safe land for all. It also affects the carbon storage because they're extremely productive environments, which means that ultimately climate change can be slowed by continuing to foster healthy lagoons across the nation. And this will then eventually lead to more favorable conditions for future generations, as well as kind of like healthier animals, plants, and humans around the globe. So ultimately, I think we kind of viewed macroinvertebrates as like a good indicator, a good, free, accessible indicator to people of a lagoon's or a body of water's health, which in turn have so many benefits that really help humans.
We have time for questions. Still some good ones.
Last time we went, we did see a large presence of algae, especially near like the ocean area, near that inlet. Did y'all see any algae when y'all were in Rodeo Lagoon?
I don't think so. No. There was a lot of sea foam, but it was mostly clear water by the ocean side.
Awesome. There was like tons of really bright green algae when we went in September. So if you didn't see that, then you probably didn't see algae. Great job, guys. That was awesome.
I have a question. Your qualitative data, right? The observations that you made. Was there anything that surprised you or stood out to you in terms of both like number of species, but also like abundance of each of the different species?
I actually thought that the qualitative data was the data that most aligned with our hypothesis because as we got to the freshwater area, we saw more species and also just more, the population was greater in general. So that actually aligned with our hypothesis.
Did you end up doing the Shannon index values?
Yeah, we did. That's how we measured the biodiversity on some of the graphs, but
Okay.
it came back insignificant. Yeah.
All right.
Okay, so our research was looking at the impact of human proximity on water, nutrient, and pH levels. So before going out to the Rodeo Lagoon, we did some research on our own to kind of understand what was already known about the subject. And we found that previous research shows that human activity does can change abiotic factors like water pH and nutrient levels. We also found out that runoff from human activity may carry excess nutrients and phosphorus into the water. This is also known as nutrient pollution. And so this nutrient pollution later leads to fast-growing algae-like bacteria to emerge in the lagoon. And this algae-like bacteria creates toxins that can be harmful to humans and animals if they come in contact with it. And it also leads to lower oxygen levels. So we kind of understood that these changes in the abiotic conditions also lead to lower biodiversity, since many organisms aren't fit to adapt in stable conditions. We also, before doing our research, we understood that pH, nitrogen, and phosphorus levels are commonly pretty good indicators of water quality in this kind of research, since they directly affect aquatic life and also organisms. And so overall, our research project aims to understand how small-scale human activity like walking paths, housing, and transportation impact local ecosystems over Rodeo Lagoon, and that will allow us to understand how we as humans can make better collective environmental decisions. So this is one of the graphs that we found most interesting when we were doing our research before going to Rodeo Lagoon. This graph shows phosphorus levels from the Chattahoochee River, and it shows the downstream levels, which is the upper line, and also the downstream levels and the downstream and the upstream. And so overall, we can see that the downstream shows significantly higher phosphorus levels. It ranges from 1,000 to about 1,800 tons. And the upstream shows significantly lower phosphorus levels. Most of them are below 200 tons. And so after about a little bit after 1990, a state mandatory restriction was put on the use of phosphate detergents. And once that restriction was put in, we saw a significant drop in the phosphorus levels of the downstream river. So this graph and this data kind of shows us how human pollution does directly affect nutrient levels, but it also shows how restrictions and environmental regulations can also reduce human impact. So this was our research location. We took 8 samples, from 2 on the human side and 2 on the trail side, and we collected 4 on each side, and we tried to space them out so that there was about 20 meters between each sample collection site. So these were some of the main some of the main biodiversity things that we noticed. So we saw that on the human side, there was more litter, more compacted soil, and fewer plant types, and the ones that were there looked pretty dry and less healthy, in comparison to the trail side, which showed greener plants, overall better soil quality, and different plant species. So this suggests that human activity does have the potential to impact abiotic factors, which also impacts the biodiversity. And overall, we did see more biodiversity on the trail side.
Okay, so our research question was, how does proximity to human activity and development affect water quality? And we were specifically looking at the pH, nitrogen, and phosphorus levels. And our independent variables were whether we were testing on the human versus non-human side, as well as the distance from the ocean. And we believed as a group that more runoff would carry more nutrients on the developed side into the water of the human side of the lagoon. And some important variables we considered were the development of the land into buildings and parking lots, as well as the presence of vehicles and their exhaust, as well as general erosion caused by human activities such as foot traffic. So the materials we used in our experiment were water quality monitoring kits, as well as water collection tubes. And we first collected 3 samples from our first site, and we mixed in a dissolvable tablet into each sample in order to separately test for phosphorus, nitrogen, and pH. And after we observed the levels by comparing the color of the sample to the picture chart. And for each test, we repeated this procedure, and we moved 200 meters further from the ocean and down the lagoon. And we repeated this for 4 tests.
And we then moved vertically across the lagoon in order to be parallel to our other samples in order to limit the distance from the ocean as a variable. And we repeated this procedure, but each time we moved 200 meters closer to the ocean for a total of 8 tests.
So for our pH data, as Oliver said, we collected 4 on the side with human impact and 4 on the side without human impact. And we saw that generally on the side with human impact, the pH seemed to be higher, which meant the water was more alkaline. And on the side without human impact, the pH seemed to be lower. And our p-value for this was 0.15. So that's a pretty low p-value, which suggests that there could be a correlation between the impacts and the pH. So our data could be statistically significant for pH. For our nitrogen tests, we found that the p-value was pretty high. So our p-value was 0.58, which means that there's not much correlation between the impacts and the nitrogen that we found. And you can also kind of see that on the graph. You can't really make out like a clear relationship between the two. So yeah. And for our phosphorus tests, we found basically the same thing with phosphorus as we did with nitrogen. Our p-value was 0.57. So there was not much correlation between the impacts, the human impacts and the non-human impacts with the levels of phosphorus in the lagoon. Yeah, so also on the graph, you can see that there's like not really much of a trend between phosphorus and the impacts.
Based off of our data that we've seen and the evidence, our new claim is that the pH of the water on the human side was, because it was consistently higher and it seemed to have a pretty high statistical significance, that the pH on the road side or on the human side is higher than that on the trail side. And we're going to go over more of why this reason makes sense in our EU, but it could be based off a lot of the reasons that Oliver had previously mentioned with the concrete and limestone runoff from roads, in addition to other human activities. Unfortunately, because of the other nutrient data didn't provide very low p-values, so we couldn't draw a lot of correlation between there. So we can't really say anything in terms of our initial claim about the other nutrient levels of the water in the human side versus the trail side. For future research, we wanted to look more at the soil quality. That was one of our initial things that we wanted to do, but we didn't have enough time when we were actually out there. And as Mason mentioned, we spent a lot of time like traversing and walking around. So for also future research, we would want to try to, again, look at where we are first and be more specific in how we can get the data from the places, because we often had to walk for a long time before we were able to collect data from places. And I think that would give us more time to look at the soil quality as well. We wanted to use more precise tools. As you guys can see, we were just dropping the tablet in and then looking at the color and comparing it to this sheet that we had, which is a very imprecise way of determining the different levels. Like it's a very approximate method that we had to use. And so for that reason, we want to, for future times, if we were going to do it again, use more precise tools that would maybe give us a quantitative value rather than a qualitative value. We would also want to take more than just 4 tests every 200 meters. Maybe every 100 meters or every 50 meters, we would take a test. Because again, I think that would help with the nutrients specifically and finding more correlation because we would have more data points. We would also want to change what we're looking at based on the tools that are available. Again, that's like similar to what we had over here. And the differences, so that's cut off a little. We wanted to look at the differences in other nutrients as well, other than just nitrogen and phosphorus, to try to further our conclusions and apply them more broadly.
Okay, so because our p-values for nitrogen and phosphorus were generally pretty high, we didn't really look at that data too much when we were thinking about the conclusions that we could draw. But because the data for our pH values was pretty statistically significant, as the p-value was 0.15, we tended to focus on that when we were making conclusions. Our initial research showed that this is sort of kind of unrelated to the research question, but our prior research showed that most coastal lagoons have a pH of 8. And so as we measured, which has to do with the salinity, so as we went from the ocean side to the side that was farther away and closer to the freshwater stream, we found that the pH actually decreased from 8 and then just like it gradually got lower. So that kind of makes sense. But then, yeah, we also found that the healthy range of bodies of water to be for pH is 6.5 to 8 in order for them to support biodiversity. So then on our graph, we found that on the roadside, most of the data points were at the max level of pH, which was 8, the max level of pH according to our tests. And then on the trail side, the data points were clustered around the lower part of the range, which was like 6.5. So yeah. So what could this mean? We sort of looked at more research after we saw our data. And then so two interpretations of the data were found. So first we found that human settlement could actually prevent runoff because low pH is typically attributed to runoff from sources like mines or landfill leachate or agricultural runoff. And so because the human settlement might have blocked the runoff coming down from the mountain, it could be that the high pH is an indicator that there was actually less runoff. And then another interpretation of our data was that limestone, gravel roads, and cement runoff from those roads could cause high pH levels because runoff carries compounds like hydroxide from limestone into the water, which could affect the biodiversity in the lagoon. Okay, so as for connections to the biodiversity EU, in the case that the second interpretation of our data was true, which means that runoff from the roads causes higher pH in the lagoon, if the pH got to be too high and exceeded 8, then it could definitely affect the biodiversity in the lagoon. As the biodiversity EU says that humans have a impact on the environment around us. And so to fish specifically, high pH could damage gills, skin, and eyes, and also reduce the effectiveness of their olfactory system, which makes it harder for them to detect their surroundings in food. So that could definitely affect the biodiversity in the lagoon if more fish were to die because of these impacts. And also as pH increases, unionized ammonia becomes the dominant form, which is toxic to fish. So basically, increased pH could cause ammonia toxicity. And so for connections to environmental justice, we sort of thought about solutions that could happen in order for the pH to be at more normal levels and less extreme. And so that could look like changing the construction of roads to be further away from the lagoon, which there could probably be like a middle ground to be found where the ease of human transport wouldn't be like too affected, but also it would be far enough away from the lagoon so that the runoff wouldn't cause the pH on the side impacted by humans to be too high. Yeah.
All right, yeah, and here are some of our work cited, and a huge thank you to Rochelle, Miss MK, and Miss Bria for making this possible. So, thank you guys.
All right, questions?
You go first. Sure. I'm just curious, did you notice if there were any pipes or like tray-looking features around the lagoon, or did it seem like all the water was coming from like the connecting stream?
Yeah, I definitely noticed
Okay.
like some large like pipes
Yeah.
that were coming, particularly mostly on the human side, but there were definitely some on the trail side as well.
Yeah. And did you, I mean, I guess this is like a whole other question, but do you think that the higher pH happened closer to any of those pipes or that kind of thing? Or is that's kind of a hard question, but.
Yeah, I think it's difficult to say because we need to know a lot of data of where the water is coming from, how it's being like, because if it's from the ocean, then that like changes than if it's being like pumped from like a freshwater facility. So we can't really like, I feel like make any judgments on how that is. And I think that's a big part of what we found is that like our argument isn't necessarily like very clear. It depends on what data you look at and how you interpret it. So yeah, I think that's like kind of a difficult thing when you're dealing with like things like pH and water quality, because it just depends on what you look at and where it's like the data is coming from.
Yeah, I would say like because our research kind of showed that like human activity could cause either like low or high pH. It was a little bit confusing, but something we did notice was that the causes of low pH were typically like stuff related to like landfills, and we didn't see much of that like on the site, and we mostly saw cement and roads, which would like indicate like causes for lower pH.
Cool, thank you.
And then my question is, I know that y'all originally wanted to look at soil and look at water quality. Do you think that the results that y'all saw from water quality would be similar if you were to test the soil, or do you think it would be different?
I think it would just provide more data points for the nutrient levels, which is really what we didn't have enough of with the water. Again, I can't really say which direction it would go until I look at the data. But yeah, the main thing that we would want to do, the soil for, was for the reason of not having not very correlated data for the nutrient levels. And it would allow us to better judge what happens to the nutrient levels, because soil is much more we can much more clearly see the different levels when we test it on the soil. So that's mainly why.
And the water is also cycling constantly.
Yeah.
So the soil might have a better indication
Right.
of long term.
Yeah. Another thing we noticed is it's not that big of a lagoon. So in that way, the water is probably, like I say, always moving around. So one side might not necessarily feel that different from the other side in terms of water, but the soil might reveal that there actually is a significant difference in the nutrient levels.
Awesome. Great. Thank you. Awesome job, guys.
All right, last group. Step on up.
No, I don't do research locations. No, I'm doing research. I guess, okay, I'll do data analysis. You were doing, now you're doing the first three. You're not doing the first three. No, I'm not doing the introduction. No, you did the introduction. All right, I didn't write them. I'm not doing introduction. I was doing the introduction. Alright, sure.
You have to start.
And then I'm doing research location.
Stop here.
I'm not just doing the introduction. You're doing that.
All right, let's give
Yes,
our
that's you.
attention to our.
Yes, it is. Because you're doing the first three. You're doing the first three. Yes, you are. Why is your name right there? No. Yeah, that's you.
All I did was raise my hand. That's the easiest.
You wrote. I didn't write the introduction. Who wrote it? I don't know. All right, sure, I'll do it. All right. Yeah, this is our presentation on how water quality affects biodiversity. So introduction to what we're testing. Our previous research includes levels of salinity, nutrients, and pH in bodies of water. These factors all determine the amount of species that can thrive. And then these parameters also influence the types of organisms in the environment. We were interested in this because we want to know what the best conditions for aquatic biodiversity are, what factors can diminish biodiversity, how different factors affect the water quality of the lagoon. We also want to know if relationships between parameters and biodiversity still exist within a singular habitat with brackish water, and then if the larger ecosystem is made up of separated microcosms, or if the species interactions are more complex. We thought it was important to do this research because we want to understand the effects of human impact on brackish ecosystems, such as salinization or increased runoff.
Okay, so our research locations, we did a total of 9 locations, and obviously there are limitations, so we couldn't do the middle of the lagoon, and so we just did the edge of it, and also we only did shallow water, because obviously you can't go deep. And then we did the lower salinity areas, which were E, F, G. They were closer inland, And then brackish water, which is the middle, include B, C, D, H. And then the north side tended to have more human activities. And then the higher salinity was A and I. And then here are some photos we took. And the research question is, what is the correlation between water quality and biodiversity, species richness, and organism abundance in the lagoon? And the independent variables we tested are distance from ocean inlet, pH, chlorine, hardness, and alkalinity. And then we measured the number of macroinvertebrates as well as the number of different species. And we hypothesized that the biodiversity decreases as minerals decrease and increases as salinity decreases.
All right, moving on. But wait a second. Okay. You might be wondering, what are macroinvertebrates? So macroinvertebrates. So macro means it's large enough for us to see with the naked eye, versus micro, you can't see it. Invertebrate means it has no backbone. So essentially, it is aquatic insects that live in streams and rivers, in our case, the lagoon. And so basically, different types of macroinvertebrates that we see can help us determine stream water quality. So, like how, you might ask again. So different species, they have different tolerances to pollution. So let's say if we find a lot of pollution tolerant microinvertebrates in macroinvertebrates in one area, that could indicate good water quality versus if you have less species of tolerance, that could mean worse water quality. So moving on to the materials we used. So we got D-net. It's a net shaped like a D that we use to collect the organisms. So we stick it into the bottom of the lagoon, shake it a little bit, and then we lift it up, let the water drain. After that, we dump it into a plastic box. We add water in it, and we count up how many macroinvertebrates we see, and then we divide it into ice cube trays with different species so we can count how many different species of macroinvertebrates there are. So we use a spoon to do that. We scoop from the little box, we put it into the ice cube tray. And then so we use also identifying sheet to see actually what kind of species it is. So it's a guide that helped us along the way. And to test the water quality, we used a water dipper. So it's like pretty long. It's like 6 feet. And then go into the lagoon, scoop some water, and then we use our special pH water quality strips. So usually it's these strips only test for pH, but this one's got like 15 different tests. We dip in the water, lift it up, and then we just compare on the side of the bottle, and we record our data. And the tests just include like pH, chlorine, nutrients, and hardness. All right, so methods. So first we had to identify the sample sites, so where we want to take the test. So all around the lagoon, from more saline sites like near the ocean to like further away towards a creek. And then we use the D-net to first like scrape the bottom of the water, the land first. We lift it up, let the water drain so there's only like mud and macroinvertebrates left. Then we flip the D-net over into the plastic box. We use the water dipper to put some water in there. So it kind of looks like that. And then we scoop it out. We scoop the invertebrates that we see into these dish trays so we can clearly see kind of more zoomed in of what species they are. So we use our sheet to identify that, and then we would record our data on a clipboard. So like this, number of macrospecies, species found. And then, using the remaining water in the water dipper, we would take one of these strips, put it in, the whole strip in the water, take the strip out at a 10-15 seconds and wait for a minute for the colors to fully up here, and then we would compare it on the other side, just kind of like this, put it out side by side, and just compare them. Yeah. And afterwards, we just dump everything back into the lagoon. And yeah, we just repeat that for all of the sites. And after data collection, we come back, we use our data regression test to see how strong our data was. So before we go into results, what can we do with the results? So the results can help environmental agencies and local governments identify kind of like areas of the lagoon that may be experiencing pollution. For example, like the north side versus the south side, or salinity stress. As we were taking tests, there were a lot of like ocean currents coming in into the lagoon. That can help better management and just protecting the lagoon in general. So NGOs, environmental, non-government environmental organizations can also use the data to help advocate for better water quality regulations. And they can do like restoration projects that can help maintain biodiversity in the more brackish middle area. And then also businesses near the lagoon, I guess like Nature Bridge, they can use this information to evaluate and minimize their environmental impacts. And it could just, in general, support more sustainable practices and compliance with environmental law. So kind of like taking steps to build a better environment.
Okay, so these were our results. We found some correlation between distance and organism abundance. So as distance from the ocean increases, organism abundance decreased. The correlation and statistical significance is kind of weak, but given that we didn't have that many data points, the p-value is probably still significant, that there is some sort of relationship. It's also worth noting that the north side had lower organism abundance, even at the same distance as the south side. So that could mean that there's human impacts that are also influencing this relationship. And we also looked at species richness. And so there's a similar relationship, though the correlation and statistical significance is weaker. Since the macroinvertebrates that we saw were small, a lot of human error in identifying the species could have resulted in the range of the variation in the data. For more qualitative data, the main species that we saw were amphipods and isopods. So amphipods, we mainly saw like scuds, and those were found closer to the ocean, but isopods were found closer to like all the sites, but most notably also in like closer inland. This sort of makes sense because amphipods are like laterally flattened, and this allows them to like swim in like tougher currents. So it makes sense that we found them in sites closer to the ocean, but isopods are more like dorsiventrally flattened, and this allows them to like bury in like rocks and more like muddy waters. So those are found in like our more marsh-like environments. And both of these species feed on like decaying organic matter and detritus matter. So they are competing for the same nutrients. So this could also explain why the places we found them were different too. Another parameter we looked at was pH, but this wasn't super statistically significant. The correlation was weak, and there just is not a lot of strong evidence to support a negative relationship. We also looked at chlorine, and this has like a p-value of like 0.85 and 0.37 for species richness and organism abundance, which is how we measured biodiversity. So there's also little evidence to support a relationship between those two. Same with hardness, although the p-value is a little bit lower, so there could be some trends, but again, the correlation is weak, and the statistical significance is not great. And the last thing we looked at was alkalinity, and same thing, the correlation is weak, and the p-value is pretty high. So we had many limitations which
we had a small sample size. We only took 9 data points, but in that we also lost a lot of data and a lot of our materials broke. So there was a lot of missing data points. Our instruments were also really imprecise. Our strips were pretty much like qualitative. We had to look at the color, and the scale of the numbers was pretty big. So it was difficult to interpret. And we were, again, only sampling from the shallows and the edge of the lagoon. So in the middle of the lagoon, that could have shown different data. And yeah, a lot of our materials broke. So we had to switch or just stop using materials, and that could lead to inconsistent data. Yeah, so for our discussion slides, some of the main patterns in our data were that the further we went from the ocean, the less the amount of species and the total amount of macroinvertebrates. And the amount of chlorine also increased as distance from the ocean increased. So our data shows some evidence that the distance from the ocean, which means that decreasing salinity, has a negative relationship with the number of species. And for chlorine, there was also some evidence of a positive correlation between increasing distance and with the chemical measure of chlorine. Our results compared to our hypothesis because it originally showed a rejection of our hypothesis because distance from the ocean would have a positive correlation with both species diversity and organism abundance. And our original hypothesis was that it would have a negative correlation since we assumed that higher salinity would lead to less organisms. But our p-values, as we saw in the graph, were 0.13 for species richness and 0.21 for organism abundance, which means that we probably don't have sufficient evidence from our graph to fully accept or reject our hypothesis. And for why we saw the results we did, we thought that organisms could have more of a desire to stay near the ocean inlet because it has more nutrients and there's more places for habitats to live in compared to the freshwater lagoon. And also the higher amount of chlorine in freshwater would cause more deaths in amphipods and isopods, which were most of the species that we identified during our data collection. And also another thing that could have skewed our results was our method of gathering data. We mostly only gathered data from the shallower parts of the lagoon as we went forward, and there might have been more organisms and species in deeper water that we didn't get to. And building off of that, there were also a lot more plants further from the ocean, and the overabundance of one plant species could be taking all the nutrients in the water, leading to less biodiversity in amphipods and isopods.
And then, so if we were to do another test, some future research improvements we could make is sample from more sites and then conduct several trials at each to limit error. So this would be like taking an average. And then we could measure water quality parameters with more precise tools instead of just like the strip, and also have a salinity tester that actually works. And then we could test salinity of the different sites, same thing. And take samples from the center of the lagoon rather than just from the edges, because that could skew it based on like what plants are living on the edges. We could like swim out to the center or something like that. And then some conclusions that we can take from our research is that biodiversity is influenced by factors like location, salinity, and nutrient levels. Though the limitations of our data collection resulted in not the strongest data, there's still some indicators. Biodiversity gradient in the lagoon was nuanced, so different species occupied different areas around the lagoon. And though all species needed to feed and survive to grow and reproduce, their different adaptations and psychological attributes allow them to survive in different environments and not directly compete with each other. And then because they occupy different ecological niches, like freshwater versus saltwater versus like brackish water, the environmental system becomes more resilient to change. As they respond to different stressors, that makes them more resilient. And then small trends in the data suggest a human influence to the presence of biodiversity, but we didn't really find anything that significant. And then human influences in this environment can disrupt the balance of like biodiversity and like species in different niches. but the small trends in our data don't indicate that much. These effects could be further exacerbated if human activity continues to expand, like if they started building up stuff on like the trail side or something like that. And then we think protection of biodiversity is important to maintain and regulate the supporting services and support. It's important to maintain and support the services of the lagoon. I don't know what that means. Yeah.
All right, I guess I'll add on a bit to the conclusion. It's kind of relates to the EU because species are all trying to solve sort of the same problems, such as like to survive and reproduce. And so many species are either adapted to like fully freshwater or fully saltwater. And so that's why like fewer species are in sort of that like awkward mid-salinity zone. And so brackish zones have like less richness in biodiversity. And then these are our works cited. That's it.
Time for at least one or two questions.
Sorry. So the other groups found more species towards the freshwater. Why do you think your group found more species towards the ocean?
I think there could have been some experimental error there. Because, well, from what I noticed, like our methods of collecting the species kind of like deteriorated as we kept doing it. Like we got a little less careful. It was like the first one we sort of paid more attention to how we're doing it and stuff.
And that happens. I know, like, there's lots of different organisms. It's definitely hard to count all of them, but that was interesting to count them less towards the freshwater.
What did you expect to find that much life, and what sort of observations did you make in terms of like vegetation change from one side to the other that may have influenced why you saw different organisms?
I mean, on the side closer to the ocean, it was more like dirt and less like grass and weeds. So we saw more like scuds and plugs there, but closer inland, it was kind of marsh-like and muddy. And so we saw more isopods on that side.
Cool. Thank you.
Yeah. No, actually.
Okay.
Okay, we're Pilar, Wendy, and Joy, and we tested how distance from the ocean in the lagoon was going to affect water salinity and how, in turn, that would affect macroinvertebrate biodiversity. How do you just kind of this one? Okay.
Sorry. Okay, so like from previous research, like we know that different species can tolerate different salinity levels. Like for example, like certain types of seaweeds and fish found in estuaries can tolerate like wide fluctuations in salinity as they transition from freshwater to seawater with changing tides. On the other hand, like coral reefs and like deeper sea creatures are like more susceptible to changes in salinity. So applying this thinking to our hypothesis, we're like interested in how like macroinvertebrate species differ due to like different salinity levels and the distance from the ocean. It's important to study how water salinity affects macroinvertebrate biodiversity because salinity determines which like species can survive and reproduce. Changes in salinity, which can be like from like climate change or like human activity, can like significantly affect entire communities of macroinvertebrates. When I was doing like outside research, I found like an article from like the National Library of Medicine that said like when freshwater salinization occurs, like these freshwater species need to use extra energy for osmoregulation, which is a process that like maintains water and like ion balance. And so when like salinity levels increase in like freshwater areas, it like increases osmotic pressure, which like significantly impacts the survival of like freshwater species. And so like most freshwater species are like eradicated once it reaches like a certain level of salinity.
Okay, so because we were testing sort of distance from the ocean and like how that affects salinity and biodiversity, we chose to just go along one side of Rodeo Lagoon, as you can see like in this map, and collect our data points on that like left side. And so how we did this is that we chose to walk 3 minutes in between each site location along the left side, so that way we could collect data that showed sort of like the salinity levels in comparison to like the distance from the ocean. And so we started at 234.85 meters from the ocean along the left side of Rodeo Lagoon, and then we walked 3 minutes along to the next site to measure salinity and count species richness and the number of organisms. And we chose these spots so that we could get like the most variety in our results and like more macroinvertebrate biodiversity. And then right here you can see the like coordinates of each of our sites and like their distance from the ocean. And on the next slide, we have some like highlights from like our sites. So we could like see like the different kinds of macroinvertebrates at each site, as well as like the turbidity of the water. And then we like have some like photos of like sort of like what part of the lagoon we're at and how far we are from the ocean. So like this was like our closest site to the ocean. And then like this one, as you can see, like we're a bit farther. And this is like the farthest point from the ocean. So yeah.
You go.
Oh, okay. So our research question was, how does the salinity from the ocean, or how does distance from the ocean affect salinity, and how does that affect macroinvertebrate biodiversity? And our independent variable was distance from the ocean, and our dependent variables were salinity and macro diversity, biodiversity, species richness, number of organisms. And our hypothesis was, as distance away from the ocean increases, salinity levels will decrease and macroinvertebrate biodiversity will increase because not many species can tolerate high salinity levels.
And then as for our materials that we used, because we're measuring salinity, we used a 4-way water meter set to EC, which is electrical conductivity. And then to get our coordinates for distance and like to see how far we were from the ocean, we used Google Maps for GPS to get coordinates, and a phone for our timer to see that we were walking 3 minutes. And then to collect our macroinvertebrates, we used a DNet, and we also used a macroinvertebrate identifier sheet to understand what we were picking up and collecting. And from there, we had them in these little like ice cube trays to count more closely and identify like what we were looking at. And then we had like a little plastic bin that all the sort of water was in, and so that way we could see like the turbidity of the water as well as just like overall like quality of it. And then finally we used Data Classroom for statistical analysis to see like what our results look like on a graph.
Okay, so how did we actually like test all these things. So starting at the point that Joy had on the map was kind of like in the bottom left corner of the lagoon, if that makes sense. But we just started there and we first found the salinity. We used the 4-way water meter set to the EC, or just like the, I think it's like something current, I don't know. Yes, that electrical conductivity. But that's just like salinity. And we set it to ppm, and we submerged the probe in the water, kind of just using the kind of methods that we learned when we were in class of how to do that. And made sure the sensors were fully covered. And then we recorded that in our data table and did it 2 more times to make sure that, the salinity was accurate for that given place. And then while we were doing that, some of our other people in the group were using the DNet to get the macroinvertebrates. So we agitated the bottom of the substrate, either using our shoe or with the DNet itself. And then we used the DNet to collect the dust that we, the dust in the water, like sandier water, that had the macroinvertebrates that were lifted up off the substrate. We removed it from the lagoon, drained the water out, and then put the contents into a bucket that had some of the lagoon water, so the macroinvertebrates would not be, just in a bucket with no water. And then, sorry, my computer just deleted all the slides, but that's okay. And then we used the bucket. We had the bucket and we took a pipette and spoon to put them into the ice cube trays. From there, we were able to see how many of each kind of species we had, and then also we identified the species that we saw. So we wrote that in our data tables. We took photos along the way. We took them of the ice cube tray and also of the location, and then we released the organisms back into the ocean or the lagoon.
And then for step 3, which is calculating distance. Joy already talked about this a little, but you want to move. We separated our sites by moving northwest up the beach after a 3-minute walk. After that, you wanted to repeat steps 1 and 2 and record all the data in the data tables. And then we continued moving up the beach until we reached the northwestmost point of the lagoon, and then we turned right. You want to walk along the lagoon parallel to Mitchell Road, and repeating steps 1 and 2. Here's some photos of us in the field. You can see over there, there's me and Joy moving the macroinvertebrates into the ice cube trays. You can see Rochelle holding some of the amphipods. The terrain was pretty tough to traverse, so we had to climb down some mountains with a couple falls. And then here's the DNet that I'm holding, where we scoop up the invertebrates. And then here is our results. This is a graph showing salinity's effect on number of organisms in the lagoon. So we kind of found that, or using Data Classroom, our data showed that salinity has a very weak relationship with the number of organisms present. You can see that the slope of the line is decreasing at a very small amount. And the p-value is 0.55, which means that this relationship that we observed is not statistically significant. And the R-squared value is also very low at 0.013, which shows that this is a very weak relationship. So this kind of led us to the conclusion that as salinity increased, the number of macroinvertebrates slightly decreased, but the relationship was very weak. So this suggests that salinity likely does not have a strong influence on macroinvertebrate biodiversity in this lagoon. We have 2 more graphs. This one is on salinity's effect on species richness. So for this one, we did find a stronger relationship. You can see the trend line in the graph showing that salinity as salinity increases, species richness decreases, and our R-squared value is greater than 0.5, which means that there is a pretty strong relationship with species richness and salinity. And the p-value is also less than 0.1, which means that our data is definitely statistically significant. So from this, we can conclude that salinity has an effect on species richness in the lagoon. And for our third graph, on distance from the ocean's effect on salinity, we found a strong relationship with the distance from the ocean's impact on salinity levels. There's a downward trend for salinity levels as you get further from the ocean. We know that there's a strong relationship because we have an R-squared value of greater than 0.5. The R-squared value is 0.552, and our p-value being less than 0.01 also shows that our data is statistically significant. And from this, we can conclude that the distance from the ocean has an impact on the salinity of the lagoon.
Maddie pretty much said everything, but like to summarize, just looking at our results, we can reject the null hypothesis, which states that like the distance from the ocean doesn't affect salinity and therefore doesn't affect biodiversity. We can reject the null hypothesis because our graph, let me see, let me go back here. Yeah, our graph here shows that the distance from the ocean and here. Yeah, distance from the ocean and salinity level has like a p-value less than 0.01, which means that there's like a strong confidence and that there's a significant relationship between these two variables. Yeah, by rejecting our null hypothesis, we have evidence that support our hypothesis that like the distance away from the ocean as the distance from the ocean increases, salinity levels will decrease, and macroinvertebrate biodiversity will increase because not many species can tolerate high salinity levels. And this graph, like Maddie said, there's a strong relationship between these two variables, which supports our hypothesis. However, we cannot fully accept our hypothesis because our data shows that as salinity levels decreases, macroinvertebrate biodiversity doesn't always increase. It shows that biodiversity only increases as salinity level decreases to its like optimal level, which is like around this area, which is like 8,100 to 8,200 ppm. So that's something that we have to consider when we're accepting our hypothesis. And also, depending on how you measure biodiversity in terms of like species richness or the number of organisms, that also affects whether you can accept our hypothesis or not, because as you can tell, there's not a strong correlation or relationship between these two variables, but there are for species richness. So that's something that you have to consider. Yeah.
Okay, so in terms of kind of just like our areas of potential error, there are definitely a lot of places where error very much well occurred or could have, whether that was due to, you know, a random error, systematic error, or just like it wasn't in our range of capabilities. So we obviously chose to attempt to eliminate bias by using a timer to walk from place to place. You know, we couldn't necessarily track our pace, and so we couldn't say that we were going the exact same amount of distance each time. And so that, you know, caused to some sites being closer to each other than others. So the exact distance between each place was not the same. And also, while we were counting the number of individual species and the species richness, we used the same process at each site. But it's possible that this process was like flawed because we had to use our best judgment to count the amount of species and to identify each one. We can't be certain that in the bin that we used to take our data, we didn't miss a species that was under a piece of algae or something like that. Or that we can't, you know, when there's upwards of 200 little scud in the water, we're not counting every single one. And so you can't really be certain that we got every single one. And then an area of systematic error that we actually ran into in the beginning of our research in the field was that our four-way water meter had like started at 100 ppm, like before putting it in any water. And so we were like, what's going on here? And so when we put it in the water, we couldn't necessarily be certain that 100 wasn't going to be like affecting all of our data, making every single one of our data points higher by 100 ppm. We do think that we fixed it before we like went on with all of our research, but we don't actually know. So it's possible that all of them were off by a little bit. So ways to minimize this potential error, we could, by using like a transect, which I put up here, we could have like measured the exact distance that we went each time. We did not think that after like looking at the transect, we were like, this is going to take more than half of our time just to like lay it all out and get to where we're trying to go. So we decided that wasn't going to be the most effective for the amount of time we had in the day. So with more time, a lot of these things might have been able to happen, but we could use transect, and we could also, go around the entire lagoon, because taking more data points is something that is always helpful in experiments. And we could have made it all the way around the lagoon, which would have let us like measure 2 different points on either side of the lagoon. So like the side on the bottom and on the top, with the same distance from the ocean. So this could have helped assure that our like species number of macroinvertebrates and the species richness were accurate, because if we took them from both sides but the same distance from the lagoon, then we would have had more data points that like could either have rejected or affirmed our hypothesis by, I don't know if that makes sense, but like we're from the same distance, and so then you can infer whether the same relationship still occurs. And then also looking back at the graphs that Maddie showed, we saw that generally as distance from the ocean increased, the salinity of the water in the lagoon decreased, but there were some outliers. And in future research, something that we could look at is like, are these like outliers pockets of salinity? What are they, like, why are they there? How do they get there? And how do they actually affect biodiversity? So like if you have like just a pocket of saltier water due to maybe there are some places where like it was kind of like a marshy, like grassy area with like water too. Like, is it getting trapped in there and stuff like that? Like, and how does that actually affect the biodiversity that we see? And then Wendy also mentioned how dissolved oxygen plays a larger kind of role in the survival of different species. And the 4-way water meter we had access to could measure pH, dissolved oxygen, temperature, and then salinity, which we chose to look into. We could have used that to also measure those different things. And that could have had a different relationship to biodiversity. And so in a future project, we could maybe look at that and then see how that affects the biodiversity that we see, as well as turbidity. We could also look at that. So yeah.
And then to sort of just like wrap up and conclude, this like really connects to our like biodiversity EU, because it shows that like every macroinvertebrate has like different adaptations to sort of like thrive in their area with different salinity levels. And like an example of this is that we found like a couple of skeleton shrimps, and they were only in areas with high salinity. And we didn't really see them anywhere else. And then in low salinity areas, we found different species that we hadn't seen in high salinity areas. And these different adaptations are like shown to like tolerate different salinity levels. And they allow like multiple species to coexist within the same salinity levels, which creates greater biodiversity. And then this means that like, the more species there are and like the higher the species richness, like the more like diverse the environment will be, and it allows them to like perform different like ecological roles. And yeah, and then also like human activities like pollution runoff or like urbanization or climate change can change salinity levels. And that could also like decrease the biodiversity, which is just something to think about. So yeah. I did have one question. So the four-way meter tested salinity and EC, and I know that EC and salinity are directly related. Why did you choose to test EC as opposed to testing just salinity? Or did you not know?
The one that we had didn't say anything. I actually asked. Yeah, I
I think
don't.
we did EC because the other one was reading really weird. Okay. And that's why.
Wait, that was like the percentage, right? That's why I was asking.
Well, there was a percentage for salt, and then there was also ppm for salt. So you might have just missed that one, like, mode. I just didn't know if you chose to do EC because you thought it would be better than choosing to do direct salinity. That's totally
I
okay.
fear that we didn't like choose that, but we just like, yeah, it was reading really weird. Like whatever we chose was like the best option for.
And like I said, they're
Sorry.
directly related, so it doesn't matter. I was just curious if there was like a specific reason that you wanted to test EC over salinity, or if it was because there were errors or something like that.
I think that's just like what we had to do because like that was like the only like option.
Yeah, when she like gave it to us, she said this is how we should do it.
Okay.
We're gonna set it up.
Okay. Is this the next group?
Yeah, that's great.
It's just.
Hi, I'm Amelia, Brooks, Giselle, and Amalia, and our project was on distance from the lagoon to test plant and soil biodiversity near Rodeo Lagoon.
Okay.
Oh, other computer, sorry.
Oh gosh, sorry.
Sorry.
Okay, so Rodeo Lagoon is a coastal lagoon that was formed thousands of years ago because of rising sea level, which created a sandbar separating it from the Pacific Ocean. It's located in Marin County, California, and it's roughly 900 meters long, 250 meters wide, and the maximum depth is 2 meters. And it was once a natural marshland, but the area was later altered by military development during the 20th century, which reduced the lagoon's size and affected its hydrology. And today, Rodeo Lagoon supports a rich but sensitive ecosystem, home to species adapted to fluctuating salinity and water levels. And because of its shallow depth and enclosed nature, it responds rapidly to environmental changes, making it an important kind of natural laboratory for studying the effects of climate change. Okay, so, Okay, last Thursday, our group conducted a field study on opposite sides of Rodeo Lagoon to explore how distance from water affects plant and soil biodiversity. And in class, we've been learning about the different types and levels of biodiversity and how they interact to create resilient ecosystems. We've also explored how human activities and environmental changes impact biodiversity using data to analyze patterns and evaluate ecosystem health. And because the lagoon's conditions change depending on location, which you can kind of see, our group wanted to understand how those factors shape local ecosystems. Using various tests to measure soil composition, moisture, and plant diversity at different distances, we aim to determine whether spatial variation influences biodiversity and to better understand how Rodeo Lagoon maintains its ecological balance. And here are some photos collected throughout our measurements.
So here is a map of our where we collected data from the lagoon. We chose two sides and we used a transect to walk back, starting with 0, 25, 50, 100, and then 150 meters back. We also did both sides of the lagoon to make sure data was accurate. Sorry. And yeah.
Okay, so our research question was, how does proximity to the lagoon affect soil health and plant biodiversity? So we were testing for independent variables was the distance from the lagoon between, like Brooks just said, so 0, 25, 50, and in increments of 50. And then the dependent variables were soil health, which was the soil pH and the soil moisture, and then plant biodiversity through species richness. And then we used a VPD gun and plant light. And our hypothesis was that closer proximity to the lagoon will have soil quality, which allows for higher plant biodiversity. Okay, for materials and methods, we used the sampling plant method, which was repeated at every location. So we placed a tape measure, which was a very long tape measure, and we placed it down on the ground and placed a quadrant over the plants at every of the same locations that we already specified. And we did this twice at 2 separate locations. And then we recorded the information down, which was using a VPD gun. And we used a value that was on the bottom right corner, but it sort of didn't really work, but we'll talk about that later. And then we didn't use the app right away, but we used an app picture this because there was no cell. So we just took pictures and we then identified it later. And then pretty much similar for the soil method, we inserted one of the moisture meters, which was a 3-in-1, and we used a sample to get the moisture reading, and we recorded the values, and it was like a 3-in-1 meter, so that gave us light, soil, and pH at every location. And then we also used a small sample of soil using this like soil core sampler, and we smeared it on a piece of paper and compared it to the Munsell soil chart to see the soil health. And yeah, this is just a picture of what the 3-way meter looked like and how we used it inside.
These were some of the different species that we found. There's a lot of ice plants, clover, sword fern, Pacific silverweed, Hottentot fig, Monterey cypress, Baltic rush, common shrubs, and coyote bush. And we noticed that there was a lot of different variety of plants in each of the quadrants as we got further along. And for the ice plant, we saw that in the first 2 tests, and later on you can kind of see how as we get farther from the lagoon, there's a lot more variety of species, but there's usually one species that's taking up the majority area of the quadrant. And then for the upcoming slide, some helpful terminology is the p-value, which tells us whether the pattern we see in data is likely or just random chance. A small p-value below 0.05 means the relationship is statistically significant. Our squared value shows how strong the relationship is between the 2 variables, and it ranges from 0 to 1, where 1 means a perfect relationship and 0 means no relationship at all.
So in this test, we tested how distance from the lagoon affected soil moisture. Our p-value was 0.58, which means the results were not statistically significant. And then our R-squared value was also very small with 0.04, which means soil moisture doesn't change as you move farther back. Although there were 2 big outliers in the group at 9 and 4, and we think those could have been by random or systematic error, like we used the 3-way meter wrong, or we just chose it in a muddy place, for example.
Okay, for distance versus pH, our p-value was 0.002, which is under 0.05. So it is statistically significant, and we can kind of see the negative correlation here. And the R-squared value is 0.0521, so therefore there is moderate correlation. And we can infer that soil pH decreases as it gets further from the lagoon, again through the slope. And there's an outlier of a pH at 4, 0 meters away from the lagoon.
This is our chart for distance compared to VPD value. And here there's also a weak correlation between the distance and the VPD, because you can see R squared is only 0.0363, which is very small. And although there is a positive correlation between distance from the lagoon and VPD value and kind of a slightly positive slope, the p-value being 0.6 means it's much larger than what it would need to be statistically significant. So therefore, there is not evidence that there's a relationship. This is distance versus total number of species. measuring kind of biodiversity. And here there's also a weak correlation with an R-squared value of 0.123. And the p-value is also 0.32, which again means it's not statistically significant and there's not evidence of a relationship.
Okay, so on to our kind of discussion. Our data showed a statistically significant negative correlation between soil pH and distance from Rodeo Lagoon, indicating that the soil becomes more acidic further from the lagoon, which supports the idea that proximity to brackish water, which means fresh water and salt water from the ocean, affects soil chemistry as lagoon spray and salt water influence the concentration of dissolved ions. And these salts can buffer pH, keeping the soil closer to neutral near the lagoon. And on the other hand, VPD and total species richness both kind of showed weak positive correlation with distance, but neither relationship was statistically significant, and soil moisture just showed no clear pattern. So those two were kind of disproved. Oh, sorry. Our initial hypothesis predicted that the areas closer to the lagoon would have higher soil moisture, less acidic soil, and greater species diversity. And while soil pH supported this prediction, the lack of strong relationships for other variables indicates that the distance alone may not be the primary factor shaping the local ecosystem. And one possibility could have been that the outliers affected the accuracy of some results, particularly in our VPD measurements and pH measurements. However, the weak or inconsistent correlations could also be due to small-scale environmental variation, such as nearby trees influencing soil chemistry, and measurement errors like a malfunctioning VPD sensor may have also contributed. And so our VPD gun wasn't really working because the air was too humid, and that slows down the moisture that the plants release. So the VPD between the water and the plant leaves and the air around them was similar. So a lot of our numbers were just like 0. And these results suggest that localized biotic and abiotic interactions play a stronger role than distance alone in shaping soil and biodiversity. Adding on, most of the sources we looked at did support our original hypothesis of better soil health and plant diversity closer to lagoons. However, the difference in our data, like in comparison to the sources, could be due to multiple localized environmental factors at our lagoons, because Rodeo Lagoon experiences high salinity fluctuation because of its kind of connection-ish to the ocean. There are variable nutrient inputs, and human disturbance could also alter the typical effects near a stable body of water. And to add on, the disturbance of soils and vegetation during the military construction likely introduced changes in sedimentation, possibly nutrient runoff and soil chemistry. And then also, because the lagoon surrounding soils are sandy and nutrient poor, proximity may not substantially improve soil quality or moisture availability. And again, this supports the idea that ecosystem biodiversity is context dependent, shaped by both natural variation and human modification.
So yeah, kind of building off Nelia, our research kind of shows that how the lagoon is very unique and hard to collect data from because of all the human impacts and the salinity from the ocean and the constant weather changes. It also showed us that it was very hilly, hard to climb mountains if you saw us up there. And it shows the lagoon has different biodiversity all around the lagoon, just because there's different species, different biodiversity, so it retains water less. Some plants and species retain water more and some less.
We also learned how it can connect to enduring understandings, biodiversity, and environmental justice. The enduring understandings for biodiversity states that humans are not separate from biodiversity, and as time goes on with human infrastructure and technology, our impact on biodiversity and its impact on us and just how we're a part of the ecosystem as well is very large. And we can see that with our research in how the military infrastructure built near the lagoon might have affected the specific sites nutrient levels or abiotic factors such as salinity, water flow, humidity, and soil saturation, which all factor into biodiversity. And because biodiversity underlies stability and resilience of ecosystems, changes in abiotic factors due to human infrastructure added, even if it was a long time ago, can alter the community structure and function a lot. And yeah.
Okay, so we just wanted to talk about some future improvements. So I think something that we could have done was gathering data in a more organized manner. So actually, like when we were taking the data, like making sure that everything was put in the right columns and rows. And then also, like as I said before, like there was kind of no like cell signal when we were all the way up in the like mountains. So having pictures of the actual individual plants printed out of like the surrounding like Rodeo Lagoon would have been probably helpful. And then the like we said, we were supposed to have like a digital version of the like manual one that we had, but we didn't have any batteries. So making sure that all of the things that we were supposed to have are like ready to be used before we start the test. And then also like having tools that might be easier to like understand and move around, because we're kind of carrying a big bag like up the mountains, kind of hard. And then also reducing biases by like placing the quadrants down based on the specific meters that we chose, because sometimes we would move around to see if there were any things. So actually sticking to the plan, and then picking locations that will actually show different species of plants, and that are not as difficult to be accessing. And then also like in a more like timely, if we had more time, we could also pick different and more data points.
So one question we asked was, how do results change after rainy versus dry weeks, or depending on the season? Because it had just rained the day before, which could have affected it, and it had kind of been wet and dry, moist all week. So testing if it was dry all week, what that would look like, or if we did it in the spring or in the summer, how that would look like. And then our other question was about testing less variables, finding out which variables we actually need to test, because it felt like we spent a lot of time testing many variables that didn't help us in the long run as much. So just going back and kind of making sure, asking that question, which variables actually will help us answer our question.
This is our bibliography, so all the sources we used for our background information. Thank you. Here's some photos.
One question? Did y'all see any changes? I know you were looking primarily at plants. Did y'all see any changes in other organisms like animals and or insects? Or did you see any animal?
Well, we definitely saw the remains of lots of birds on the right side. The left side was a little more dry, so I feel like I don't think we saw.
Yeah, we didn't
really
see many besides the spider that went into our bag.
Oh.
But yeah, nothing much besides the species.
And I'm sorry you didn't have batteries. I gave them to her. I guess she forgot.
It's okay.
It's okay. Great, thank you. Yeah, good job, guys. All right, last group, you're up.
Okay. Hi, we're the soil and plant moisture and proximity to water group. So we tested how the distance from the lagoon impacted the soil or the plant moisture in proximity to the lagoon. Yeah. And I'm Audrey, Logan, Sean, and Simona. Okay. So, introduction. What is already known. We know that soil and plant moisture tends to decrease with increasing distance from the water source due to the reduced groundwater access and the higher rate of evaporation. We know that soil texture strongly influences water retention, as finer soils hold more moisture but may limit the drainage. We know that proximity to coastal lagoons can create a unique salinity gradient in the ecosystem, influencing the biodiversity, vegetation types, and the adaptations of the species living there. We know that past studies in coastal wetlands close to this one and dune systems, such as the Point Reyes and Rodeo Beach areas, show that small-scale topographic changes can have major effects on plant and ecosystem structure. So why this interested us is that we're interested in exploring the relationship between topographical features such as water and its effects in regard to plant function, resources, and other ecosystem processes. And we're also interested in how this research connects to the physical geography and the plant ecology, showing how even a subtle shift in elevation or soil type can change the biodiversity of the ecosystem. So the importance of this research is that by understanding these relationships, we can help predict how ecosystems like the lagoon ecosystem may respond to climate change, drought, or human water management and interference. And our findings could possibly inform restoration and conservation efforts, especially in the sensitive coastal habitats where water availability drives vegetation health. Okay. So this is our research location. We chose the eastern bank of the lagoon to minimize the ocean and the human activity effects. And we sampled every 60 meters, I wasn't there, but they sampled every 60 meters along this line here that allowed them to capture how the distance from the lagoon affects the soil and plant moisture without losing the gradient effect. And the layout here can show sites 1 through 7 moving away from the lagoon towards higher ground. They use GPS coordinates to ensure consistent spacing and accuracy. And the map also displays topography, water proximity, and nearby human features like trails and the art center, which they took note of as they collected data as well. However, it was not obviously one of our tested variables. So yeah, I'll give you guys a second to look at the map, but kind of everything.
Okay. So in designing our research question, we wanted to keep our question broad with a general direction of what we wanted to investigate, but also something that could be pivoted on the day if necessary and if we felt it needed to be. So as a result of that, we settled with the question, how does moisture in plants change depending on the proximity to the lagoon, either in change of elevation or distance? And then our independent variables were distance from the bank and elevation, and then our dependent variable was the moisture content in both plants and soil. And then plants would be measured by a VPD gun, while the soil would be measured by a soil moisture meter. And then finally, our hypothesis was that soil and plant moisture would generally decrease as distance and elevation from water sources increase. Okay. So once we got to our location, we decided to keep our question and hypothesis as it was, since we didn't feel like it required much more specificity. And to keep it general, would be to keep it a bit more true to the broader ecosystem rather than a specific part or function within the broader location. And so the materials we used were the soil moisture meter, the VPD gun, the transect tape measure, the compass app to measure altitude, and then a quadrat. And our procedure involved, as Audrey mentioned, first measuring the 60 meters from the previous location in as straight as a line as possible. And then once the location was marked, we would randomly place our quadrat in the area. And we would do this by having a member hold out a quadrat and spin their arms left to right. And then while another was facing away and would say stop randomly. And this removed bias, but also increased randomness. And then we measured the VPD of plants by assessing the plants closest to the corners of the quadrat, because our quadrat didn't always include plants. And so we wanted to keep that lack of bias by picking plants closest to the four corners. And this would give us a range of plants in each location while also giving us plants to study. Like I mentioned, often our quadrats were dry and barren, and so our assistant Jeff helped us and proposed that alternative solution. So this way we could still collect data about plants living in the area while not increasing bias. And then we measured soil moisture using the soil moisture content by measuring moisture
by measuring center of the quadrat, and then we used the Compass app to measure altitude. These are just some images of how we collected data. As you guys can see, sometimes the quadrants weren't completely flat. Like you can see here, we're hanging our quadrat on like a little bush thing on the side. But this just increased randomness and eliminated the bias of us patting it down to the plants we wanted to highlight. And the photos below were of Sean carving through the Marin Hills with a transect tape to measure out the next 60 meters in each trial. So yeah.
So here is the one graph of our results, which is a scatter plot graph comparing distance from lagoon in meters to average VPD or vapor pressure deficit. With soil moisture here is the Z variable, just for additional information. Generally shows that as distance increase, average VPD decreases, meaning that moisture increases. Right, the regression line here are our squared value, or the proportion of variance explained by the model is 0.185, which is relatively low in terms of being 18.5% of the variation explained by the model. But our p-squared value is also low, although it's not quite low, 0.05. But there is some confidence in this relationship between the two models. Even better, and for altitude, again, a similar scatter plot graph comparing altitude in feet to average VPD. Again, similarly, average VPD is going to decrease as altitude increases. Our p-value is lower here, suggesting a greater statistical relationship and confidence in terms of the relationship between the variables. And our squared value is slightly larger, explaining more variance, but still not very high. There's another static plot graph comparing distance from lagoon to soil moisture. Again, generally showing that as distance from lagoon increased, that the soil moisture generally got more dry. We used a digital moisture meter, so it was a categorical value as opposed to a 1 to 10 for a three-way moisture meter. But given how high our p-value is, there is little statistical significance of our graph. Similarly, in terms of comparing altitude to soil moisture, again, soil moisture is generally going to get more dry as altitude increases, but our p-value is also relatively high, meaning there's little indication between the variables. In terms of discussion, as the graph shown, average VPD decreases as distance from the lagoon increases. For example, at 0 meters, VPD was generally hovering about 1.9 kPa, which is pretty high relatively. But as distance and altitude increased, VPD generally decreased to being about 1.6 kPa by the end. Again, for VPD, our p-values were relatively low, meaning there was a statistical significance between our variables. But our squared values were also low, meaning there is a lot of variance that is not explained by the model due to sources of error. In general, this decreasing VPD means that moisture increases, right? So transpiration is reduced and moisture increases as distance from a water source and altitude increases, which rejects our hypothesis, which I will get to. In comparison, if VPD increased, that would mean that stomata would get smaller and CO2 uptake would be reduced and transpiration would increase, meaning that moisture would be reduced. In terms of soil moisture, as shown in the graphs, soil moisture generally decreased as distance and altitude increased. For example, past around 70 feet in elevation, 100 meters away from the lagoon, soil moisture is generally dry plus, as our additional moisture meter showed. But given how high the p-values were, there's little significance or confidence in terms of the relationship between the two variables, and we would need more data to confirm with our hypothesis. So our data for VPD generally rejects our hypothesis, as hypothesis would assume moisture would increase, which means, moisture would decrease as elevation and distance increased. However, moisture increased, meaning VPD decreased, which rejects our hypothesis, which is due to a variety of factors, which I will get into. In terms of soil moisture, ultimately our data was inconclusive, and we need more data, whether it supports or rejects our hypothesis. In terms of variables, VPD is, or explaining the p-values or statistical significance of our data. VPD is generally dependent on variables more directly correlated with the independent variables we chose here, so elevation and distance. However, sources of error likely resulted in this rejection from our hypothesis. BPD is generally dependent on humidity, air temperature, temperature of leaves, and elevation, among other factors. In comparison, soil moisture is dependent on more factors than just distance and altitude, which we look at here, including precipitation, temperature, soil texture, elevation, plant species, and land use, which gets into our potential sources of error. In terms of the VPD gun and our tool use, I think source of error could have occurred from where we actually pointed the laser of the VPD gun on specific plants and the plants themselves, as well as other factors such as air temperature and weather. For example, is fairly foggy and cloudy out, which could have affected our data, and just plant adaptations in general. Again, as Logan will point out, our distance was non-linear. Our transect tape was not in a straight line, which could have also affected our data in terms of true distance from the lagoon. In terms of measuring altitude, our compass app was not necessarily effective, as it only measured in tens of feet and was often contradicting itself. Again, weather, terrain, and location likely affected our data in terms of temperature conditions, precipitation, slope, organisms, and soil texture. And we had various inconsistencies with plant species in terms of their adaptations, for example, root length and what moisture would be available to them. And also our data set was fairly small, which could explain our variance.
In terms of future research, I just want to reiterate a couple of points of error that Sean pointed out, but then also get into solutions that we could have used to remedy the inaccuracies for a future research opportunity. So if we were to do this again, we would probably have to find a different way to measure out 60 meters. Like Audrey said, we tried to take our best guess with making sure we were mapping as straight as we could. But unfortunately, just because hiking through the terrain was very difficult, Sean often just picked the path that was safest for him to walk through, which led to a windy path. So instead, what I think we could have done was twofold. One, we could have tried to make the transect tape a little more taut and try to make as straight as 60 meters as possible. But secondly, we could have brought a rangefinder instead. This way we could zap exactly how far Sean was away from us. And in this way, we could figure out how far away we were actually traveling. And while it may not have been exactly 60 meters, we would get we would make our data more accurate. And we would still get our 10 values that we needed. In terms of studying the plants, in terms of the plants we selected, with our experiment, we mostly floated between shrubs, grasses, and leaves. And in the future, I think it'd actually be more meaningful to just study one plant, because it's hard to say how each plant responds differently. It kind of actually does add towards our experiment because we're studying how plants also shift as we travel and how their anatomy shifts how they hold water. But in the future, perhaps as a secondary experiment, we could study a singular plant. And something probably most notable about our experiment is that we often threw a quadrat down with basically barren land. The entirety of the quadrat was basically dead. And we wanted to be able to study living plants that would give us a more accurate VPD value. And so what that left us with was our corners method. But I think what this also caused us to do was literally cherry pick, because we were hand picking the leaves that we wanted to study. And whether that's, a bit of bias or not, I guess is kind of up for interpretation, because it is a systematic method, but there was possible room for bias. So in the future, we would want to find a solution that would be able to perhaps factor in the dead plants that landed within our quadrat. And then finally, like Sean mentioned, we couldn't study altitude very accurately, only because since we had no cell reception, our compass app was really guessing. The telltale sign was when we were on the bank and it said that we were 10 feet above elevation. So we knew that the compass app was doing its best, but it wasn't actually perfectly accurate. So in the future, we would want to find a better method of measuring elevation accuracy. In terms of questions that would have been beneficial for us to answer, one of them is what plants generally hold more moisture, which hold less, and then how do the species change as distance from the lagoon increases. Again, this basically helps us understand how, when we're taking the VPD, how much the lagoon is in fact is a factor versus the plant anatomy itself. In addition, what is biodiversity like based on plant and soil moisture as we back away from the lagoon? Really a similar line of thinking. How do plants change? How do ecosystems, like micro ecosystems within the ecosystem, change? And then finally, what is the biodiversity like based on, how do plant species change as distance increases? Only because we want to study, again, how plants shift and also how they hold moisture and how that shift occurs. So in terms of our conclusion, we noticed that since VPD increased as we traveled away from the lagoon, biodiversity generally takes place a little differently as we travel away from the lagoon, because different plants exist that rely on different systems. Some rely on themselves to hold in more moisture, while others rely on the lagoon. And what that leaves us with is a broader ecosystem that's less susceptible to like catastrophe. We live in California where droughts are often a risk. And if water were to significantly decrease in the lagoon due to a drought, the entire Marin Headlands ecosystem would not break down, because there are plants that rely on less moisture in order to survive, but also to keep their fellow organisms alive, whether it's local animals or insects. In addition, humans have a role on protecting these ecosystems, because we have a role of not causing that drought, but also promoting or making sure that our studies are also not hurting these ecosystems. I guess when we were carving through the mountain, we were damaging some plants. We were stepping on plants. And in the future, I think we want to figure out a way to minimize the harm we were doing on the actual ecosystem. Yeah. This is our bibliography and our acknowledgements. Thank you. Oh, yeah.
Great, good job, everyone. I know that class is over, so just want to say thank you so much for all of the effort that you put into your projects. Y'all were an absolute pleasure to work with, and best of luck on all of your